With this panel, it's really going to be an incredibly wide-ranging, universe-trotting panel discussing life for us and the future of life, not only here, but across the universe. And we're going to start first on this planet, looking at the concept of time and health, and ask Esther Dyson, who's going to build on knowledge brought out of 4 years of looking at 5 different communities in a 10-year project, The Way to Wellville, understanding how to improve health and what the meaning is for that and how we can possibly do that. So what are some of your learnings,
Great.
Esther?
So first of all, how many of you were here at the AI session this morning? Okay. One of the wonderful things about DLD is how different parts get connected. And it turns out AI and health and human beings and life are intimately connected. So I was listening to the AI session this morning, and they were talking about consciousness and what is it. I'm going to come back to that. And later you're going to hear Petra talking about life. And life really requires 4 functions. Reproduction, it's got to keep making itself, and she'll talk much more about that. And metabolism, it requires information, the DNA, so that when it reproduces, it can reproduce the same thing rather than something else. And it requires a container to keep it all in one piece. And I would argue that consciousness inherently includes a sense of the past, the present, and the future. You can construct something in your mind, and we'll talk about that more. So now I'm going to go back to myself 4 years ago, looking at the tech world, trying to figure out what would be a little more useful than Uber for shoe shines and games. And I started looking at healthcare and wondering, why do we spend so much money on healthcare, fixing something that's broken, instead of investing in health itself and keeping people in good shape? There's a wonderful book by Nicholas Taleb called Anti-Fragile. The basic idea is, if you're healthy, stress makes you stronger. Exercise is stress. It's stress that makes you stronger. Being exposed to a lot of pathogens improves your immune system. Resilience, it's a wonderful thing to have, but you have to invest in it by providing the right amount of stress, but also allowing for recovery. And that's fundamentally what we're failing to do. And I'm talking more about the United States. Here in Europe, there's a much greater sense of social investment, but even here, I would argue, we're not doing it perfectly, but I'm just going to take blame for the United States and consider that I'm not talking about Europe because I don't speak for them. So, decided to start a project to work with 5 communities to help cultivate health rather than improve healthcare. And the idea was to do it in a community where it's people around you, it's the institutions, the parks, the schools, the food you find, a community that takes care of children, whatever. It sounds really nice and sweet, but it actually requires business kinds of thinking, responsibility, judgment, accountability, doing things at scale. So from that initial idea and thinking about health sort of in general, smoking, cancer, diabetes, maybe prenatal care. 4 years later, it's now a 10-year project. 4 of the same communities, one that switched out because that community wasn't really leaning into the project, and much more focused on the children and the parents. Because what we discovered is it's one thing to fix things that are broken. To create them healthy, you really have to start at the beginning. And that ultimately means the parents and the children. You fix the parents, it's nice to fix the parents, but the actual return is in healthier children that are resilient. So as I was doing this, I started learning more and more about addiction and neuroscience. And here's what I've learned. Our project is fundamentally, I was trying to explain this to some people at an AI conference. Our project is fundamentally about fixing the training set for young brains. If that, as science people prefer that to childcare. It's those habits, those instincts that you learn as a child that then follow in the rest of your life and make you resilient not just to physical disease, but also to mental disease. Because what I believe is that addiction is not about drugs. It's not even about cell phones. It's fundamentally about short-term thinking. It's looking for immediate gratification, immediate pleasure, rather than longer-term purpose and agency. People who become addicted lose their sense of the future. They lose their sense of the past. In a sense, they lose that long-term thinking that underlies consciousness and a sense of agency. How many of you have heard of the marshmallow experiment? Okay, you give a child a marshmallow or some other wonderful sweet, and you say, don't eat this now, just sit here, and in 10 minutes or 15 minutes, someone will come back with a second marshmallow. The children who eat the marshmallow right away, they're short-term thinkers. In later life, they're more likely to drop out of school, to become addicted, to be unemployed, to end up in jail. And it's not because those children are stupid or bad, it's because they learned from their experiences, if you see that marshmallow, take it. It might not be there in 10 minutes. The examiner might not come back. And I'll spare you a lot of the long-term details. But this is fundamentally what's now underlying our thinking, that the childhood environment is key. It's not that easy to fix. It requires childcare. It requires loving adults. You can't put all the children in an orphanage with bots. You have to somehow, again, figure out how to fix the parents. And so we're working, I'll go into this later with anybody who's interested, but this fundamentally is what we're trying to do. And as we look around, we discovered, in a sense, the people who invited us in, they too were addicted. They were addicted to pilot programs, to short-term fixes, rather than beginning at the beginning when children are born with training for the parents in some form or another, with loving grown-ups. But also, who else is addicted? Investors who invest for the short term, businesses, politicians, all of us. And all of us need to start thinking longer term. Because when you get back to what life is about, short-term thinking is addiction. It's staying alive. It's doing what is required immediately. Long-term thinking is purpose and building for the future and investing in your children. Thank you.
So from health and time, we'll now move to Petrus Willa, who is at the Max Planck Institute here in Munich, to speak about the creation of life. It sounds a little bit like Frankenstein. What is the minimal representation of life? What does it look like, and how do we create it?
Yeah, thank you very much. Yeah, so this was actually a very fascinating introduction. Yeah, so ever since I was a kid, I always wondered what there was the difference between the living things and the non-living things. And I really thought I should try to get to the bottom of this. And I actually studied physics rather than biology because I thought that, well, biology is studying life, but if you really want to get to the bottom of things, you probably should go and do the fundamental physics way. So I made my career really not with this question. You probably can make a career in science with this question. but with developing methods how to study molecules in cells. And if we look at cells, cells are really the smallest unit of life. That's basically the outcome of the cell theory of the 19th century. So today, it's basically accepted fact that all living things are made of cells. So if you're thinking about the minimal representation of life or any form of life, it's a cell. And also, and that's a very important part, every cell comes from another cell. It was actually a big thing to find that out, because in the old history, people thought, philosophers and the first scientists around, that cells or living things could be, coming out of mud with a spark from somewhere. And it was actually a big accomplishment of science to find out that this is not the case. That if there are cells coming from something, there were already cells in there, like mud or so. But that immediately brings another problem out there. Well, can't be true entirely, because there must have been a first cell, and that first cell must not have come from other cells. And that brings us immediately to the question of the origin of life. Well, there's a lot of research going on the origin of life, and Dimitar will probably also connect to it later. But we know nothing about it. I mean, we don't find any fossils about the very first cells. What we can speculate is that they are pretty much similar to today's cells in the way that they have compartmented. They have a shell around them, probably made of something similar to today's cells, which are membranes. And there's probably some information unit inside the first cells that was pretty similar to today's RNA. We don't know it, but there's a lot to be, to speak for this hypothesis. But the question is now, can you really come up with something like a very first cell? Could we make something like that out of things that were not cells? And that's our research that we want to do. We want to basically build cells, minimal cells, first cells from parts. And of course, we cannot start with atoms, because that would take us too long and we're not good chemists. So we start with functional modules. And of course, we are cheating a bit. We're taking these functional modules from existing cells, because we're not really interested in the very modules. We are interested in the very principles of the modules. And what that means, I will show you in the, well, no, that's a slide that has sneaked inside. Why is there not so much research on that? Because if we're asking this question of the minimal cells, well, what biomedical research is all about is it wants to have physiological relevance. So biomedical research is funded well if it is very connected to the cells in our body, because then we can assume that there's something good for our health coming out. These questions that I ask, origin of life, first cells, they're very academic. That's the reason I'm at the Max Planck Institute, which is the fundamental research institution in Germany. So at the same time that we know more about the fundamental principles of life, we of course lose physiological relevance. And that's just a problem for this kind of research. This is the reason why probably a lot of people who are super interested in that might not be able to do it. And I have to say, I mean, this is just a side mark. Here in Germany, we're in a really good position to be able to do this kind of fundamental research. Out there in the US, in the UK, the big research hubs elsewhere in the world, they don't have all that much money to do that kind of research. So I really speak for Germany for that. But now get into the real stuff. So what is it about? What are we looking at? Well, Schrödinger, who was also a physicist, he made a really good comment about what should be at the very bottom of any living system. And this is the ability to make order and sustain order out of energy, which is against the second law in a way. So you have to not invalidate it, but you have to use it very efficiently. So this energy that we take up, that organisms take up, needs to be very highly ordered. And the nutrients that we live on are very highly ordered energy. So with energy, you can create order and you can sustain order, although nature would like to have everything in disorder, as you might know from your rooms. So first of all, if we want to come up with something that looks like a living system, it should be able to do that. Make order out of disorder. Sounds a bit mysterious. Can we do that? Well, cells can do it. And this is a cell that is a simple cell, super complex compared to the first cells, but simple compared to the cells in our body. It's E. coli, it's a bacterial cell. And this cell has a very peculiar way of dividing. It basically oscillates proteins between the two poles. It's basically growing only in length, and after some time, it finds its middle and assembles a ring in this middle, and that ring contracts and makes two new cells. But how does that cell find the middle? By these oscillations of the proteins. And so we started with this. We wanted to reproduce these oscillations. And we took the proteins out of the cells, only 2 kinds of proteins, and brought them with ATP, with energy, on a super simple membrane that mimicked the inner membrane of the bacteria. And as you can see here, they self-organize. These proteins, 2 proteins only, they self-organize and make patterns. And of course, they don't make patterns, they are not moving directionally. What they do is like a Laola wave in a football stadium. They basically bind to the membrane and they go off the membrane, and they do it basically coordinated, like the people in the soccer stadium doing the Laola wave. They stand up and sit down, and these molecules are pretty much doing the same. And because they do it coordinately, it looks like a wave. So it's kind of a brain in this reaction between the molecules. It's kind of funny. It's a very simple It's a very simple chemical reaction. And we brought these proteins in a box that looked like a cell, and they do exactly what they're supposed to do in the bacteria. So not much in the bacteria itself, you have about thousands of proteins. But only 2 of them are necessary to do this very important part. And of course, now the next question, and that's a very big one, is can we also assemble this ring, which is supposed to divide the bacteria? And that's what we are working on at the moment. We have already accomplished to reconstitute, to make these rings, and they are actually dynamic, as you can see in the movie on the right-hand side. This is just the ring protein. It's just one protein with energy, and it does that. And if you bring all these 3 proteins together, the ones that do the oscillations and the one that makes the ring, you can see in this little red-green part, you can see that these oscillations are indeed able to position the ring proteins in the middle. And that's a super simple system, and it does already something very, very fundamental. So we are in good, we are on good track. We are far away from anything living. But we could show that with very small pieces, you could actually go pretty high in complexity already. And that's what I wanted to present to you. Of course, that's not only my work, that's a teamwork, and that's my group. Yeah, thank you very much.
Thank you very much. So now we'll move on to Demetrius Sasselov, who is a Harvard professor of astronomy, to speak about finding life. Mae West said, is that a gun in your pocket? Are you happy to see me? I can report that Dimitar is carrying a spaceship in his pocket. So watch out, Donald Trump.
You just revealed my secret. Well, it's an honor for me to be on this panel with such amazing people like Esther and Petra, and thank you for the nice introduction to what I'm about to say. I'm the astronomer here, the stargazer, so let me try to put this into the cosmic perspective. And first, I want to tell you a little story about 8 years ago when I first came to DLD, and was nicely introduced, like always, by our amazing connector, Steffi, to the legendary Hans-Ulrich Obrist, who in his very customary way asked me a lot of questions, as he always does. And he asked me a very simple question: what is the future of astronomy? Normal thing to ask an astronomer. And I quickly answered to him, biology. Biology is the future of astronomy. He looked puzzled, and then I realized that I was a little bit too provocative maybe in my answer. But what has happened now, 8 years later, I'm back here because the future has happened. Biology has become now the future of our discipline, astronomy, and many people are now involved in doing this. So I want to use this opportunity to just give you one example of how what Petra is doing is actually the first step in us finding a very important answer to the question, is there life elsewhere beyond Earth? And even if we all agree that yes, there is, the question is how? How do we do this? And now it will be nice if I could have my slides, so I can start that. I have the clicker, but no slides. So I'm going to use one particular example, and that particular example is the moon of the planet Saturn, Enceladus. And our expectation that there is actually a very active ocean under its icy surface, and we might be able to sample this very contemporary and active Earth-like ocean for life.
Could we try and get the slides up?
I do really need my
get the slides up. One of these slides is actually only released a week ago, a week-old picture of Saturn.
Yeah,
is that correct?
yes,
So we'd
but
love to-
Do you think that clicker might do
no, that's my
No,
own
they don't have
clicker.
the slides are not there.
I use it for a different.
Sorry.
Okay. I think we're going to have to go without the slides, it seems.
No slides. Okay.
Oh,
I
okay.
sent them a couple of days ago, but
Alas.
they didn't.
Picture,
allow.
if you will,
Yes.
Saturn.
I wanted to pay homage to the amazing Carolyn Porco, the leader of the imaging team of the Cassini mission for Saturn, who was here at DLD 10 years ago to show you the first pictures. How many of you were here and saw those pictures of Saturn from the Cassini mission? That was 10 years ago. Unfortunately, she couldn't come today, and so I was going to show you some of the newest pictures from Cassini. Many of them involve not the beautiful planet Saturn with its rings, but that particular very exciting moon, which Carolyn had earlier shown to have geysers, active geysers, which constantly producing a plume of water. Of course, once these geysers emerge above the surface of the moon Enceladus, the water freezes, but anything that this water carries is there. That includes salts. For example, we know the salinity of the water that comes out from the geysers. It's like similar to that of the Earth oceans. We know that there is some gases which come out together with the water, like hydrogen and methane, which is extremely interesting because hydrogen is a very good feedstock molecule for any life, and methane as well, but indications of processes that we know are similar to that on Earth today and have been in the past. And so the question was, could we fly through those geysers and see what's in them? Cassini did that a few times, very close to the surface, literally kilometers above the surface. But Cassini, which, by the way, is a beautiful spacecraft that was the size of this whole stage, did not have any of the instruments to tell us whether there is any other evidence for possible life in these geysers. Nobody had planned for this in the beginning. So it left us with this big mystery. What's going on? Apart from knowing...
It looks like we have the slides now.
Oh, we do?
Fantastic.
Okay.
Well done.
Thank you.
Bravo. Thank you.
Okay, so very quickly, thank you. Very quickly through the beautiful images, I do owe it to Carolyn to show you some of them. This is what you've seen. This is Saturn backlit. The sun is behind the planet Saturn, so you see all this beautiful light coming through. And this is the amazing Carolyn, who is the leader of the imaging team for Cassini. And this is the moon Enceladus, about the size of the British Isles, a little bit bigger than Germany. And there are the geysers on the South Pole. And when Cassini flew through them, of course, it was flipped over like this. As Esther would tell you, in space, up and down doesn't matter. And so in close-up, there they are. Beautiful geysers. You see they come from strings of valleys, which are cracks in the ice mantle of the satellite. That's a very close-up of one of those valleys. Cassini flew literally kilometers above it. Even closer up, you can see individual boulders the size of these cubes here on those slides, and you can see exactly where the geysers are. As I told you, they come directly from the subsurface ocean. We know that because Cassini made enough measurements to tell us that this ice crust is very thin, especially there where the valleys in the South Pole are. So whatever water is pushing those geysers directly comes to the surface. So, as Carolyn would tell you, if there were microbes in this ocean, it will be raining microbes on the surface of Enceladus. Frozen, but as you know, microbes can be frozen and we can still tell that they're microbes. So the big challenge, of course, is what do we do about it? How do we do it now that Cassini has actually bid its farewell? As you know, Cassini was a long-lived mission, but had to be discontinued. This happened just a few months ago in September. This is the last image of Saturn from behind, from pretty far away, just before Cassini was plunging into the atmosphere of the planet Saturn. To the upper right corner of this image is Enceladus, just above the limb of planet Saturn. And now you will see how Enceladus is going to disappear behind that limb of Saturn, literally minutes before Cassini plunge into the atmosphere is no longer there. So that's the last view of Enceladus for us as well. And the question is, what do we do next? NASA had already 2 proposals for similar large missions like Cassini to go to Enceladus and tell us what's in these geysers from the point of view of life. Unfortunately, they were not well prepared, according to NASA, and NASA last month decided that they're not going to get funded. They're big things. They would take years, decades, to actually get there. This is going to happen eventually, but we are talking about 30 years from now. So in the meantime, is there something we can do about this? Is there something smaller, faster, and cheaper that can do this for us? And so this leads me to look into my pockets for something. And as it happens, I have a satellite in my pocket, a working version, which is right here. And the person who is responsible for that introduced it just an hour ago here at DLD. Zach Manchester, who is right there, the amazing Zach Manchester who builds those things. How many of you were at Zach's panel an hour ago? Yeah, a few of you that were there. There is Zach. There is his satellite. So one application, they were talking about saving Earth. What about detecting life in the solar system? This thing, or better, a box of 100 of those, can get to Saturn in 2.5 to 3 years. Not many, many years, as Cassini had to, because it was big and heavy. This is not big and heavy. It can get there very fast. Of course, the challenge is it cannot do very much. Even 100 of those will have to be very clever about deciding what exactly is it going to be evidence for life so that we can send the next big Cassini to actually prove that. And so here we have to have the big question, which Petra is already working on. What are the smallest components that are specific to life and we can identify as biotic as opposed to something which naturally could occur? And so let me give you one possible example. One possible example are the membranes that were in Petra's images of those little cells. We believe that you cannot have life without membranes and compartments. And those little bubbles, or as we call them, vesicles, are made of molecules which are called lipids. They form bilayers. They look like this, strings of carbon atoms attached at one end to 2 oxygen atoms, and they like water on one side and hate water on the other side. So they arrange themselves. They have the ability to self-assemble, exactly what Petra was talking about. But they're also simple. So if you do spectroscopy, maybe you can measure what is in those in that water from Enceladus, and tell whether you have a biotic distribution, which indicates maybe there is life there, as opposed to abiotic distribution, which we know these kind of molecules occur naturally out there. And the way you would do it is by sending 100 of Zach's chipsats, the little things, and having 5 of them, for example, have laser beams shining, going ahead of the other 95. And then you have little spectrographs which intercept those laser beams behind them as they're passing through the plume of Enceladus and tell us, is there something there that is interesting or not? And one day, we'll have to convince ourselves that we have to send something more expensive and bigger there. But why not do something faster, cheaper, and better for the time being before we actually spend a lot of money on a single experiment which will go there? And the big question there is we don't know exactly how to do it. And the challenge is for people like Petra and me to work with others to actually find out what these units are. And if we do, in the process, we'll also help with what is happening down here on Earth. And hopefully, we can pass it along to Esther and have a better life on our own planets. Thank you.
Fantastic, with the next space mission to Enceladus. Esther, maybe bring it back around in the last few minutes we have here.
We actually have 10 more because they started at 35, and so
okay.
we should get 10 or 15.
Okay, we'll have a little bit of discussion. Then we'd love to take questions from the floor as well, but just to sort of kick it off, if you could round the discussion a little bit here. We started with, with yourself, sort of health and time, moved to the fundamentals of life, then the finding and identifying of life. How does this bring it back to the physiological relevance of us here today and how this ties together?
Yeah, well, actually, I'd almost go in the other direction, because what each of them is saying is that life is not about substance, it's about physics, about process. And in a sense, life depends on reproduction, which is a physical process mediated with this information and these other things, but evolutionarily, it created, life by definition has the purpose of reproducing itself. Mm-hmm. And that purpose ends up becoming, when it's infiltrated with time, a much broader, longer purpose that creates human beings. And what that means, in a sense, whatever kind of life gets created, it will inevitably evolve into something with a longer sense of time, or it will die off immediately. But in a sense, evolution is not done. We keep evolving because the things with purpose keep living longer. The things without purpose eventually are going to kill themselves off. And this is the fundamental question creating humanity, not can we solve environmental problems, but do we have the purposefulness to solve both environmental problems, to wander into space, to find new forms of life, and fundamentally to keep ourselves alive rather than succumbing to that short-term purpose that technology has. We're getting so good at creating things that are bad for us but pleasant in the short run that we're killing ourselves off if we can't develop that sense of purpose.
So, Petra, let's bring the question of time into your work and your thinking. A lot of what you spoke about, pardon me if this is the wrong way to express it, but it's sort of the big bang of life. That foundational moment of life. So add that element of time to your work, and that brings us to the next phase of evolution, or what is the next step?
Well, I would be hesitant with talking about the next step. We need to find out the laws for the first step. So what is about life that has this purposeness on it, right? So what I think needs to be formulated, and it's actually a question to the fundamental physicists, also theoretical physicists, how can you formulate purpose so that it's a scientific formulation? I guess everybody of us has a sense for purpose, right? But what does that mean? It's the same about entropy, right? So 150 years ago, everybody knew, the scientists and also everybody else with common sense knew that things get messed up, right? There's some sort of inherent, tendency of matter to get disordered. If you release a gas, it will the particles of the gas will be everywhere with the same distribution. So everybody knew about that. But only after the formulation of the concept of entropy, we had a physical understanding of that. So we have no physical understanding of purpose, which is basically the big divide between the sciences and the humanities, because the humanities have a very good understanding of purpose, very good definitions. I hope so. I'm not a humanities person myself, but I assume that they have. Also consciousness, right? But if we talk about life scientifically, we need to get to the point to talk about this quality of life, because everybody knows that there is this quality. I think it's no point to deny it. I have my fellow scientists very often say, well, that's not science, right? We're not talking about purpose. That's not science. But you cannot talk about the difference between life and non-life if you don't bring that in. And I think it's a fundamental challenge. It's a fundamental scientific challenge. And I would really, I'm not a theoretical person myself. I mean, I did some theoretical physics in my studies, but I think that's a huge, task, theoretical task also to be solved. I can only deliver the experiments.
So, Demetrius, are these questions of purpose and life spoken about in terms of, in your circles, in terms of alien life forms that we're looking for?
Yes, of course. In fact, that's exactly the cosmic perspective to that question, is once we understand how a system like that emerges, how life origins are, we have a chance to understand the distant future of life. And the distant future of life is astronomical in the sense it's cosmic. We now know, in the last 2 years, we know for certain that there are many places which resemble the Earth in the galaxy and beyond in the universe, our universe, which then begs the question, if the chemistry is the same everywhere, which should be, then there should be multiple origins and life emerging in different places. And then what is our place in that? We don't know now, but part of it is because we don't know what it actually takes. In other words, we don't understand our own origins. And then the sense of purpose comes next. Once we understand that, you will have the cosmic perspective, because you already have done the physics or the astrophysics part of finding where those environments are. And we know there are many of them. So that's the cosmic perspective to the same question.
How much is there a risk that we have a biological bias, which is that the things we're looking too much for things that resemble us as opposed to different?
We have that bias for sure. I mean, that's the only way in which we can do it. We have one example, and anything that we do is, whether we admit it or not, depends on that one example. But that's why you need for Petra to succeed in the lab. Because then we will know the answer. It is an experimental thing as opposed to a philosophical question. By philosophizing, we'll never have the answer. By building it, by having working examples, we'll know the answer. And then we'll finally detach ourselves from the bias, or at least understand the bias, and know what is wrong with our bias. Now we don't.
It may not be philosophical, and it may not be experimental. It may be a question of basically modeling, looking at all the elements and molecules and seeing,
right,
are there other combinations of molecules that actually could produce some of those same phenomena that we've seen, carbon and...
Right. There is actually a chemist, biochemist, who is also a computer scientist, and I asked him, why don't we just model it in a supercomputer? And he showed me that you cannot do it with the current supercomputers. No, so that's why the current supercomputer is Petra's lab.
Yes, but it is a great challenge for AI and for nanotubes and quantum computers.
We'll come to that point eventually, yes.
So bringing that back to human health and our learnings here, how would you relate the findings and the way forward?
Well, I mean, the fundamental problem is you cannot take that motivation away and still have life.
So it goes from a biological item to a community.
Yeah, and well, forget the community part. Just, if you made a person without desire, they wouldn't live.
Right.
And what it means is any other form of life is going to have that same challenge. If it gets sufficiently good, it will get good at satisfying itself too easily and may lose its purpose to live long. I don't know, but it's, I think ultimately addiction is an inherent part of life, which is kind of scary and sad.
Which could be how the first life forms came about, with their addiction to growing or multiplying.
And they didn't have enough stuff so that they would grow too fast and kill themselves.
I would love to open up to a quick question or two from the floor, if we have any. Do we have a microphone? Quick question. May I borrow your microphone quickly?
Self-service.
Hi, my name is Maximilian Schich. I'm an art historian. The question is, if you talk about life versus non-life, at some point the question of complexity comes up. And you can reduce entropy by local activity, and it's not quite life yet. So the key question is, should there be something like an integrated science that integrates the humanities and the natural sciences, that deals with all these processes, like complexity science tries to do, in order to put us into a very, very long story, since the Big Bang, in one story, and also puts us in the place where we are indeed rare. Like if you read E.O. Wilson, for example, has this, he says, yes, life may be abundant in exoplanets, but we are actually life forms that are eusocial, we are altruistic, and we're the only one on Earth that does not do so instinctively, but actually consciously, which is really cool, and also rare, and may actually make our fragility more precious, or something like that. So the question is, do you see something, how that can be done outside of the current siloed university system where it is not possible?
Quickly, at the Max Planck Institute.
Or the Santa Fe Institute.
Yeah, so I don't think we need another science, another discipline. I mean, I think we need a good talking between the disciplines, and that's already what's going on, right? So there are initiatives everywhere, basically coming up now that where the physicists and the chemists and also the philosophers talk together. So of course there need to be a new agenda in a way. And I think we are pretty close to this new agenda. And complexity, you're totally right, is a fundamental term. We have not found yet a very good definition of complexity, by the way. And I think we need to be more precise if we talk about it.
Yeah, I want to emphasize the point which Petra just made. That's one of the challenges in accomplishing this, is that you need active collaboration by people from several scientific disciplines that have not collaborated in the past. And my experience, we've had this team now at Harvard for 10 years, is that it's very difficult to do. It's very difficult to convince the people in those different departments to work on this together, as opposed to just clap and wait, or worse.
You may already have read this book if this is your interest, but I would recommend the book Scale by Jeffrey West.
Yes.
That talks about
complexity.
It's a physicist who looks at society and biology.
Fantastic. Thank you very much. With a round of applause now for our panelists, as I think we're running a little bit over now. Thank you very much for a fantastic discussion, which we can continue off stage.